Hello everyone, welcome to this channel called Learn Bio with Janet. I'm teacher Janet. Today we'll be discussing Techniques of Answering Biology, Paper 3 of the KSSM Syllabus. So this is a new paper, Paper 3, which is a practical paper, and students will have to go to the laboratory and carry out experiments for the various science subjects. So let's find out more about this paper and how to tackle the question and do the experiments. Let us look at the format for this science paper 3, which is called Ujian Amali Sains Bersepadu or Integrated Science Practical Test, right? So it is a practical test that's carried out in the laboratory, and it consists of questions for 3 subjects: biology, physics, and chemistry, all given together to be done in the laboratory, one after the other, for a period of 2 hours 15 minutes, okay, the time. So our code for biology is 4551/3, right? So a question will be asked on biology and 1 question for physics, another one for chemistry in the paper, in the test paper. Now, type of item is that it is a subjective item consisting of structured questions. So the word item here means questions. Now, number of questions, there are 3 items of questions, 3 questions. We answer according to the subjects that we register for. If the students register for the 3 subjects, biology, physics, and chemistry, they will have to answer the questions for these 3 subjects, right? If they only choose 2 subjects, then they will answer the questions for those 2 subjects. Now, so the 3 items include biology, paper 3, chemistry, paper 3, and physics, paper 3 questions. Okay, let's talk about biology, just biology. Total marks is 15 marks per subject, okay? Same for chemistry and physics, 15 marks each. And the question is divided into a few parts. So method of giving response is by writing the answers in the exam paper, and then you hand out your exam paper. Now, duration of exam per subject. So let's talk about biology. You will be given 5 minutes to plan, check the apparatus and to plan for the experiment. Do some planning for the experiment. And then the time to answer the question is 40 minutes. So all in all per subject is 45 minutes. So if you are taking 3 subjects, then your time will be 3 times 45, 2 hours 15 minutes. Okay? So when you go into the lab, you can just imagine how envision or imagine how it is done. You can visualize how it's done. 3 sets of apparatus will be given to you in 3 different trays usually, and then you have to check it first. So let's look at a sample of the schedule for the first session. Okay, it depends on which session you're in, but let's just take the first session on that day of the exam. So at 8 o'clock, the exam starts. Now, then you're asked to check the list of apparatus given for the 3 subjects. So since 5 minutes is for checking and planning per subject, that means you have 15 minutes, 3 times 5, 15 minutes to check your apparatus and to plan your experiments for the 3 subjects. After that, at 8:15, then only you start the practical exam. Okay, so you'll be asked to start. Ready, get, set, go. Then you start the paper. All right, just joking, So it's like a race in a way, because you have to plan your time properly, and the time allocated is only 2 hours 15 minutes, no more than that, right? After that, you have to hand up your paper. So answer the 3 questions. Now here I give you just a sample timer for you to take note. Let's say you start at 8:15. Now you start trying out the biology question first. You do the experiment. By 8:55, that means 40 minutes after the first, after that, after you start the experiment, start doing the questions, you should have ended. So 8:55, okay, and the first practical. Then you go on to the second practical test for another subject. So add another 40 minutes. By 9:35 you should finish. And then after that, the third practical, another 40 minutes. So that will add up, the time will add up to 10:15. So by 10:15 you should finish the last practical. Okay, so it's good to take note of this. Just a basic idea of where you should finish, because you need to time yourself, or else you may spend too much time on one question and then don't have enough time for another question, right? So plan your time properly. All right, now, so take note of this time. It's just like a time to let you know to go on to the next subject if possible, the next question for the next subject, right? So at 10:15 a.m. the exam will end for the first session, and then students will have to go to the quarantine room, okay, like going to, within inverted commas, jail for a while, so that questions don't leak out to other students until 2:30 p.m. Then you are released. Okay, right. So I hope this is clear with you all. Now, if you have any doubts about anything, all right, please do confirm with your teacher, all right, and get updated, since this is the first time that the paper 3 practical exam is being carried out. The paper 3 integrated science practical test is being carried out. So get the updates from your teacher and confirm everything. If you are not sure, please confirm your teacher. Don't just be in doubt. Okay, so everything here, what I've stated here, please do confirm your teacher. All right, our main aim is to go into the question, to try the sample question. So for each question, for example, biology question, you have 5 minutes to check your list of apparatus and plan your experiment, and 40 minutes to carry out the experiment and answer the question. So basically the total time will be 45 minutes for each question, right? And same for physics and chemistry. Each question is 45 minutes. Now, you can choose which subject or question you want to start answering first. Which question you want to start answering first. There are 3 questions, 3 main questions. So you can choose whether it's physics, chemistry, or biology you want to start off with. So my suggestion to you is start with the question that you have confidence to do first. The easiest question for you. All right? So that you have more time, so that you have time to do the other 2 questions. You'll be able to do it fast. All right? You start with a difficult question, then you may get stuck somewhere, and then there'll be not enough time for the other 2. And to build up your confidence, so do the first question, which is easier, the easiest question first. There's no need to follow the order of the questions. So the student must be wise in allocating time to answer each question. Make sure you answer within the 40 minutes for each experiment after you have checked your apparatus. So there are 2 types of questions that will be tested in paper 3. Question with a guided experiment, question without any guided experiment. Now again, for all this, please confirm with your teacher, right? So I'll just explain to you what has been informed so far, and please confirm with your teacher for further updates and so forth. So for the question with guided experiment, it will include instructions and procedure for carrying out the experiment. So it's easier to do this question. The student carries out the experiment according to the procedure and then answers the questions. So 2 out of the 3 questions in the paper will be guided. But there's one question, it can be for physics, biology, or chemistry, we don't know, that is without any experiment, non-guided assignment or question. So no procedures are given, and the student will plan his own experimental procedure based on the objective that is given in the question paper and the apparatus and materials given. So what's the objective of the experiment will be mentioned. Of course, it has to be mentioned, or else you don't know what experiment to do. Then you have to find the MV and RV there, and then looking at the apparatus and materials, you decide what procedure can be carried out, all right, using these apparatus and materials, right? So the student carries out the experiment and answers the questions based on the results of his experiment. So I've often been asked whether we need to memorize the procedures in our biology experiments. The procedure, kaedah. Of course, you have to memorize, because in case your biology question is the one that is non-guided, that means you have to plan your own procedure, right? So the tip is to memorize the apparatus and materials and procedures for biology experiments, especially for the short ones, short experiments. Okay, like that can be carried out within 10 to 20 minutes, right? That can be carried out in a short period of time. Okay, and leave you time to answer questions. So examples, okay, let's look at some examples of these short experiments. Osmosis experiments, right? Enzyme experiments, and then experiments related to finding the energy value of food samples, right? And so forth. So please memorize the procedures and the apparatus and materials for different experiments. So the format for the practical science paper 3, right? Or science test, or ujian amali science, is that there are 3 main questions or items. One question divided into a few parts for biology, 15 marks. One question for physics, 15 marks. One question for chemistry, 15 marks. Total marks is 45 marks. So the important part is this one. What do we have to answer in the question paper? So the questions in the question paper, for example, for biology, will test your science process skills, whether you have these science process skills and whether you can use them, right, to answer the questions. So they are called the SPS, science process skills. They are mental processes which promote critical, analytical, and systematic thinking. Okay, examples are observing. So in the paper, it will say, it will state it this way: state the observation for the experiment. One observation, for example. And then the second skill is measuring and using numbers. As you are carrying out the experiment, you'll be measuring the volume of water, weighing something to measure, or to get the mass, weighing something to get the mass of a sample of food and so forth. Then inferring, okay, in the question paper, it will be state the inference for the observation, okay, which is actually an explanation or the reason for the observation. So we'll come to that in a short while, and this is the important part that you have to train yourself to be able to do, to carry out these skills. Hypothesizing, that means state the hypothesis for something, for the experiment. Controlling variables, you have the manipulated variable MV, responding variable RV, and the constant variable CV, or the fixed variable. They would know how to control them when doing the experiment. Communicating by using graphs and tables to show your results, right? And interpreting data, maybe interpreting data. The data is from the graph. For example, you have to find the relationship, state the relationship between two things, and then after that you may have to give explanations. Defining operationally, this is also important skill which many students are weak in. So we're going to find out how to give an operational definition for a term. Predicting, okay, this can come out as a HOTS question. Predict what will happen if a certain situation is given, when a certain situation occurs. So you have to apply your science, your biological knowledge to answer that. Okay, can come out as HOTS question. Classifying, meaning that we group the items into a few categories, right? Experimenting and using space and time relationship. Now, how to do well in paper 3? How to score and get 15 marks or close to 15 marks? You need to try a lot of questions on paper 3 and learn the technique of answering the questions. Okay, learn the science process skills. Right, so visualize that you are in the lab and then we are doing this experiment, and then we'll learn the techniques of answering the questions, okay, to make sure that you become skillful in answering the questions for paper 3. So first of all, when you go into the lab, you'll be given 5 minutes to check your apparatus and plan your experiment, to check the apparatus and materials. So let's say you're given the list here, and in the tray you have all these apparatus and materials. All right, so look through it, because the apparatus and materials will tell you how to give you an idea what experiment to do, how to carry it out. So firstly, you are given a beaker with 100 ml sucrose solution A, another beaker with 100 ml sucrose solution B, and another beaker with 100 ml sucrose solution C, right? So tick it up if they are there. So all these solutions are of different concentrations. Now, number 4, 2 potatoes are given. And then there's a forceps. Number 6, stopwatch. 7, cork borer to produce the cylindrical potato strips. Ruler, white towel, electronic weighing scale or electronic balance or digital balance, and then a knife, filter paper. All right, so tick is up here, and then plan your experiment. So you need to read the question. Right, so let's try out a sample practical question for biology. Total number of marks is 15 marks, and the time is 40 minutes because 5 minutes has been used for checking the apparatus and materials. Okay, let's read the question. You are required to carry out an experiment to investigate the effects of different concentrations of sucrose solutions on potato strips which are 5 cm in length. A. Using the apparatus and materials provided, plan your experiment. The experimental procedure should include the following steps: method to handle the 3 variables, precautions to be taken, and other steps. Then write your procedure below, right? So to start off, first of all, find the objective or the aim, because from there you're going to determine your MV and your RV. Okay, determine the manipulated variable and the responding variable. So if you look at the beginning of the front part of the question, carry out an experiment to investigate. Now this is where you get your objective, to investigate or to determine or to study or to compare something. The effects of different concentrations of sucrose solution on potato strips. So this is your objective. Underline it. Next, determine the MV and the RV, the manipulated variable and responding variable from the objective. Okay, when you come across the phrase to investigate the effects of something, that something is the MV. To investigate the effects of MV on RV. So your MV are the, can you tell me the MV? Concentrations of sucrose solutions. The different concentrations. Okay, that's your MV. And what is your RV? So here it's only stated potato strips. So what aspect of the potato strip are you going to measure for RV? So you have an electronic balance there, electronic weighing scale. So it tells you that the mass can be weighed, the mass of the potato strips. So your RV is the mass of potato strip. All right. Now, what about your constant variable, the CV? Now, the CV is not the length of the potato strip, because this, it doesn't mean it has to be exactly 5 cm in length. Actually, we are measuring the mass for the RV, so this is not actually the CV. The CV or constant variable is the, what is it? Time taken to immerse the potato strip or to soak the potato strip in the sucrose solution. Time taken to immerse or soak potato strip. Okay, so you have to fix the time. Now, since the time for your biology practical is only 40 minutes, you cannot soak it overnight, right? Then you won't have any results to write down here. So soak it for about 15 minutes or at the most 20 minutes. Okay, because after that you need to get the answers, and then you have to also answer the questions, right? Okay, so now it's time to write out the procedure. Okay, now you can refer to your, if you've forgotten about it, you can refer to activity 3.5 on page 63 of your textbook, right? And read through the procedure first, try to memorize it, and then write it down. Make sure you know the method to handle the 3 variables. Okay, that must be stated in the procedure. All right, you can pause the video and write your experimental procedure, and then after that we will look at the answer. So basically what we're going to do here is, first of all, we're going to take the potato, and then we are going to use the cork borer and push it inside the potato. Okay, After that, you take a little, there's a tiny rod that's attached also that you can use to push out the potato strip, cylindrical potato strip. So produce 3 cylindrical potato strips, right? And after that, put each one on the white tile, measure 5 cm and cut it with a knife to make sure that they are of uniform length, so that it's easier to carry out the experiment. Next, wipe the potato strip dry. Very important precaution to ensure more accurate results. Next, weigh the potato strip. Weigh each potato strip, right? And record the mass. So you should prepare the table ready to write in the results, and don't just scribble on any piece of paper. Okay, right. Next, soak the 3 potato strips in the 3 beakers of water for 15 to 20 minutes. You decide. Okay, so let's say we soak it for 20 minutes. Now you must start the stopwatch here. Start the stopwatch and then put all the potato strips inside and soak it, 1 in each beaker of sucrose solution. Use the forceps to put the potato strip into the beaker, right? Next, after 20 minutes, take out the potato strip and again wipe it dry with the filter paper or tissue paper, and then weigh it again to get the final mass. So you have initial mass and final mass, and that's it. Okay, that's the procedure. Here's the answer for question A on the procedure. So 3 beakers labeled A, B, and C are filled with sucrose solutions A, B, and C, which have different concentrations respectively. So actually the solutions have been given to you in the respective beakers, and you do not need to transfer it anywhere else. But anyway, this is written down to show us that the MV is handled by having sucrose solutions of different concentrations. Okay. Next, using a cork borer, 3 cylindrical potato strips are cut. After that, they are cut into cylindrical strips, and then after that, using a knife and ruler, each strip is cut to be 5 cm in length, as we have seen in the previous slide. The 3 potato strips are wiped dry with filter paper and weighed individually. So wiping it dry with filter paper is to get rid of the excess water, and that will make the measurement more accurate. So this is a precaution, right? Now, the initial weight or mass of each strip is recorded. So that is related to your responding variable. You need this value. Okay? So remember to weigh the initial weight of or mass of the potato strip. If you miss out this part, then you can't get the results for your experiment, right? Now, the 3 potato strips are immersed separately in the sucrose solutions A, B, and C for 20 minutes. Then ensure that each potato strip is fully immersed in its sucrose solutions. So this is another precaution. We can write 2 precautions down, all right, but at least 1 must be given. The stopwatch is started after 20 minutes. The strips are removed from the petri dishes and wiped dry. Okay, so the precaution is mentioned again, but actually it's the same as the number 3. Now, the important thing is that 20 minutes is stated here. So this is how you handle your constant variable. You fix the time for soaking or immersing the potato strip to be 20 minutes, right? This must be mentioned. Next, each potato strip is weighed again to obtain the final mass of the strip. So that is your responding variable 2. RV1 is the initial mass, and RV2 is the final mass. The data is recorded in the table, and the percentage change in the mass of each potato strip is calculated. So this is the responding variable 3. Actually, it is the main responding variable. I call this the main responding variable. Okay, the final responding variable that we want to obtain through calculation. Okay, and I will consider the change in mass and or the final mass of the strip as the sub responding variable that we find in the lab, and from there we calculate the main one, which is or the final responding variable, which is the percentage change in mass. Okay, now let's say you have done the experiment and now you have tabulated your results. Okay, so this will test your skill of measuring and using numbers. Now on the left hand side of our table, we write sucrose solutions of different concentrations. Unit is m or molar, right? And then you have the sucrose solutions here, ABC, but we do not know the concentrations, right? It's only through the results that we can determine whether they are hypotonic, hypertonic, or isotonic solutions. That's all. So initial mass of potato strip in grams, and then the final mass of the potato strip in grams, right? For A, from 2.30, the final mass becomes 2.15. So from the initial mass of 2.30, 0.30 to 0.15, there's a decrease in mass. For B, initial mass is 2.28 grams and final mass is 2.51 grams. So from 0.28 to 0.51, so there's an increase in mass. And lastly, for C, 2.31 to 1.98. So from this value 2 to 1, there's a decrease in mass, right? Okay, one more tip is that please get ready, drop the table neatly on a clean sheet of paper, right? And do not write your answers on a dirty piece of paper or tissue paper or filter paper, which may get thrown away, get lost, or fly from the table. So you write this down on a clean sheet of paper. Sheet, S-H-E-E-T, not S-H-I-T. As students are very fond of saying that. Okay, now let's see the questions. Second question, B1, carry out the experiment. State the observation for the potato strip in sucrose solution B, 1 mark. So this question tests the skills of observing, your skill of observing and stating the observation. So let's look at the results here again, right? Now try and write down an observation. So before you write down observation, please take note that an observation should have 3 main things. What are the 3 main things? It should be from the table of results. Should be the MV, the sucrose solutions of different concentrations, and then the initial mass and final mass. Actually, the observation has to do with the final mass, but we can also write down the initial mass. So your RV is the final mass here. You have to state, okay? And you have to state the value, right? The reading, you have to state the reading, okay? But take note that it's for sucrose solution B, so it's the second one here. So write down an observation. Pause the video and write the observation yourself. Test yourself. And then look at the answer. Okay, pause the video. Right, so let's look at the answer. In sucrose solution B of a specific concentration, we don't know what the concentration is. The final mass of the potato strip is 2.51 grams. And as a side note, the initial mass of the potato strip is 2.28 grams. Right? So the main observation is on the final mass, actually. Okay, the main observation is about the final mass. So let's look at the components. What are the components of this answer? You must have MV, all right? So your MV is stated here, the sucrose solution of specific concentration, right? And then your RV is the final mass of the potato strip. And the reading is 2.51 grams. Please have the units too. Without units, it will be wrong. This component will be wrong. Okay? So MV, RV, and reading. And the RV, of course, is the one from the table. It is the sub-RV, which we measure in the laboratory. The main RV is the one that you get after calculation or that is stated in the objective. Okay? Sub, SUB, sub-RV. So take note, observation should have MV, RV, and reading with units. Now, this initial mass can also be written down. Okay, so it's for the sucrose solution B here. This one. Don't give the wrong observation for the wrong sucrose solution. Right, second question, B2. State your inference for B1 or explain your observation in B1. Okay, so this question tests the skill of inferring. Now, what is an inference? An inference is an explanation or initial explanation or reason for an observation. So you can use the word because. Why is this observation like that? Because then you have to explain using your biological knowledge or concepts. Okay, so these are the results of the experiment. And let's look at the tip here. For inference, you have to compare with the other observations, see what's the difference. Okay, whether the result is higher or lower, okay, etc. And then after comparing, you give a reason for the observation. You can use the word because, right? So looking at our results here. Now, take note that is the observation for in B1, which is related to the second sucrose solution, B. So this set of results. So if you look at the results here, we'll see a difference. For sucrose solution A, the potato strip loses mass, right? For sucrose solution B, the potato strip gains mass. And for sucrose solution C, the potato strip loses mass too. And loses mass, I think more than the first one, as we will see through our calculation later on for percentage change in mass. So we are focusing on B. Now try and write your inference right now on a piece of paper. So pause the video and then I will discuss the results after that. Right, let's look at the results. So in sucrose solution B, the final mass of the potato strip increases or becomes higher because becomes higher after soaking. So this part is just to lead you to give the reason. So there may be no marks for this part. There may be or there may be no marks. So let's look at the reason, which is very important. Now, so this is related to the concept of osmosis, right? So why did the potato strip in B gain mass, right? Actually, it is gaining water, right? The water has diffused into the potato strip by osmosis. So it's because the sucrose solution B is hypotonic to the cell sap of the potato cells. So this word hypotonic means it has a high water concentration but low solute concentration. Okay, low concentration of sucrose, the solute. So hypotonic or dilute lah. But here the right term is hypotonic. Use the word hypotonic to get your 1 mark. And you must mention cell sap, not just hypotonic to the cell. Okay, it's hypotonic to a fluid in the cell, and that fluid is the cell sap. Okay, if you don't write the word cell sap, you may not get the mark. Now, water diffuses into the cells. Okay, not out of the cells, into the cells, and then the process is osmosis. From a region of higher water concentration outside the cell to a region of low water concentration in the cell, right? And this increases the mass of the potato strip. That's why it became heavier after soaking in the sucrose solution, right? So 1 mark for each of these parts. So this is the way to explain anything related to the process of osmosis. First state about the sucrose solution or whatever solution is used. It could be salt also, whether it's hypotonic, isotonic, or hypertonic, okay, to the cell sap or to the cytoplasmic fluid, okay, of a cell. Usually for plant cells we use cell sap. Then later the next thing is state what happens to the water. Where does it diffuse? Into the cells or out of the cells? And then by osmosis. The process must be stated. And thirdly, what is the effect of this on the cells, right? So this increases the mass of the potato strip. Now, next question is construct a table and record all the data collected from this experiment. Your table should have the following titles: sucrose solutions of different concentrations, initial mass of potato strip and final mass of potato strip, and then percentage change in mass. So this tests the skill of communicating, right? So first of all, looking at the titles that you have to write in your table, write down the units at the side here. Sucrose solutions of different concentrations. Unit is M, right? Or mole per decimeter cube. And then initial mass of potato strip in grams, and final mass of potato strip is also in grams. Percentage change in mass is in percent. Write it all down here so that you don't forget to write it in the table. All right, next you have to construct your table. So there are 4 different columns for you to write, to fill in. So draw up a table with 4 columns, and then since there are 3 solutions, so 3 rows, but plus the titles, it'll be 4 rows. Okay. Now try and write this, draw up this table yourself and fill in the readings and do the calculation. Okay. So pause the video and try and drop the table. So for you to be good in all these skills, for example, in drawing tables, you have to practice drawing the tables for biology experiments. So practice makes perfect. Okay, pause the video and try the question yourself. Right, let's look at the answer. Right, so we have the 4 columns here. Sucrose solutions of different concentrations, M. Initial mass of potato strip, G in grams. Final mass, grams in grams, and percentage change of mass in percent. Here we must know the formula to calculate the percentage change in mass, which is final mass of potato strip minus the initial mass divided by the initial mass, not divided by final mass, divided by initial mass, then multiplied by 100. Okay, right. So the sucrose solutions, we don't know the actual concentrations. We just write A, B, C. All right. So this is the MV, right? And then here's the data that we have already recorded earlier from the experiment. And then the percentage change in mass is calculated. So here I write down the calculations first and then give the results or the final answer. So how is this table marked? Now first, if you have the titles, all these titles, and the units are complete, all right, you will get 1 mark here. So make sure that the units are written in. If you didn't write the units for 1 column, for 1 title, then you will lose 1 mark here, right? Then the second mark will be for the correct data, the data that is written down, okay, and that you obtain from through the experiment, okay, through doing the experiment. And then finally, the last column is the calculation, so there's 1 mark for calculation. And please show the working. Okay, so here final mass for the first one, first solution A. Final mass is 2.15, so minus 2.30, the initial mass, divided by the initial mass, 2.30, times 100. So use a calculator and the answer will be negative 6.5. Okay, so the negative sign must be there because 2.15 minus 2.30 will give you a negative value. And then for B, similarly, final mass is 2.51 minus 2.28 initial mass divided by 2.28 times 100, you get positive value of 10.1. Okay, and then the last one, okay, work it out yourself, and you should get negative 14.3. Right. So for all these values, please take note that your decimal place must be consistent, right? For example, for the initial and final mass, you recorded it according to 2 decimal places, right? 0.30. So for all the 3 values here, it must be all to 2 decimal places. You cannot have one 2.3 and then this one 2.28. So if you just write 2.3 is to 1 decimal place, and then the second one you write 2.28, it's already to 2 decimal places. So it's not consistent, and you will get the recording of data here wrong, right? You will lose a mark here. Similarly for the final results here, 6.5, so it's to 1 decimal place, and so all the answers here are to 1 decimal place. Okay, be consistent to write down the values or readings or answers to the same decimal places, okay, for each column here. Next is a question on classifying. Now, the potato strips were immersed in the sucrose solutions for a while. So classify the potato cells in solutions A, B, and C into the two different conditions, that is either as turgid cells or flaccid cells. Okay, so highlight here, turgid or flaccid cells, right? Turgid or flaccid cells. Okay, right, so how are we going to classify? Now, one way is by drawing a flow chart, another way is table. So I would suggest you use table, as this is the normal way to classify the something, classify the items. So draw a table, and on one side, the left side, you write turgid cells, on the right side, flaccid cells. All right, now try and answer the question. Pause the video for a while. All right, let's look at the answer. So turgid cells are the cells in solution B. Why? Because there is it has gained mass, it has gained water, so the cells will be full of water, and then they'll become turgid or firm, and full of water. The flaccid cells are those in the potato cells in solutions A and C, which have the cells have lost water to the surroundings, right? So the mass has decreased, so the cells become flaccid, right? So 1 mark for each. Lastly is a question on predicting or prediction. So this can be a HOTS or KBUT question, which tests your thinking skills. A boy is bitten by a leech. His mother puts some salt on the part where the leech is attached to the boy's leg. Predict what happens to the leech. So this boy is bitten by a leech and can't come out. So the mother puts some salt on the part where the leech is attached to a boy's leg. Predict what happens to a leech. So if you have been unfortunate enough to get bitten by a leech, now this can happen if you have gone into, let's say, to an area like near a pond, somewhere wet, like paddy field or pond, and then you're not aware that sometimes the leeches will start to latch onto the leg, attach itself to the leg, and then start to suck the blood. We call it linta, right now, linta. So it's very hard to pull it off. Okay, so this wise mother put some salt on the leech, okay, the part where it's attached to the leg. So what happens? And then explain your answer. So prediction is 1 mark, explanation is 2 marks. Okay, think for a moment, write down a prediction and your explanation. Pause the video. Try it yourself. Right, let's look at the answer. So put on your thinking cap, as they say. Try to think. How is this related to osmosis? So here is our answer for the prediction question, the HOTS question. The leech dies and drops off the boy's leg when the mother puts the salt on the leech. So this is 1 mark. Now how to explain? How does the leech die? So the explanation is similar to the one that we gave just now, in the sense that we have 3 parts to the answer. First, you mention what type of solution is found outside the cells. So is it a hypotonic, hypertonic, or isotonic solution? In this case, the salt forms a solution which is hypertonic to the cytoplasmic fluid of the leach's cells. It's more concentrated. Okay, the salt solution is concentrated with solutes, with salt. So it draws water out from the leach's cells, right, by osmosis. Now, note here that we don't say cell sap, hypertonic to cell sap, because cell sap is only for plant cells. Okay, so for animal cells, we use the word cytoplasmic fluid. Now, water diffuses out of the cells by osmosis from the region of higher water potential in the cell to the region of lower water potential in the salt solution. So it's down the concentration gradient for the water. Okay, the water molecule moves down the concentration gradient. And then the leach loses water and becomes dehydrated. Its cells will shrink and it becomes cremated. Okay, and then it dies. So this part is 2 marks, so they may pick any two. But I think the first one is very important to mention that the salts forms a solution that is hypertonic to the cytoplasmic fluid of the leach's cells. Right, so that's all for this lesson. Now in the next video, the second video, I will continue to explain some more questions about osmosis, like operational definition and hypothesis. So it's a short video, okay, for the second part. And then in the third video, I will discuss another experiment, for example, the vitamin C experiment, so that you get some practice, some more practice for your paper 3, because practice makes perfect, right? You must practice applying your skills, science process skills, to answer the questions. So that's all for this lesson. Thanks for watching, and please share, like, and subscribe to my channel. Goodbye for now.